Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Precision Matters: The Critical Role of Accurate Liquid Handling in the lab. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the Comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Artel by Advanced Instruments, who developed the content for this presentation. Advanced Instruments is a global company offering a novel portfolio of analytical tools, including volume verification systems that provide NIST traceable results down to 10 nanoliters. Osmotech, a robust line of osmometers to support bioprocessing and quality control, and Solentum, a portfolio of best-in-class imaging and cell single disposition technologies for cell line development workflows and assurance of micro-osmometers for regulatory bodies. Now, with that out of the way, it's now my pleasure to welcome our speakers to today's webinar. And first, I'd like to introduce you to Andrew Herder. He is a field application scientist at Advanced Instruments. There he is. Hi, Andrew. With a wealth of experience in liquid handling automation systems for high throughput laboratory workflows. Andrew specializes in integrating robotic platforms and liquid handling technologies to streamline sample processing, improve reproducibility, and accelerate research timelines. His work supports a wide range of applications from genomics and drug discovery to clinical diagnostics, where precision and efficiency are paramount. And next, I'd like to welcome Candy Gilman. She is the Education and Training Manager. There she is. Hi there, Candy.
Hello, Sonia.
Hello. Manager at Advanced Instruments, where she leads the company's global training initiatives on liquid handling quality assurance. With over 25 years of experience in technical product training, Candy is recognized authority in pipette quality management and pipetting proficiency training programs. Now that we got the introductions out of the way, I know that it's going to be Andrew up first, but before I pass the controls over to Andrew, we do have our first poll question. So Andrew, I'm going to launch that poll question now. And audience members, you should see it appear right there on your screen. And there you go. So the question is, what keeps you up at night? Okay, is it quality and compliance issues, or is it efficiency issues, variability between devices and or operators, or cost issues, or is it all of the above? So go right now and cast your vote in the poll question there by just clicking on any of the answers that you see there. And then we will leave this open for about 45 seconds just to make sure that we get everyone involved in this poll question. Then I'll shut the polls down, share the results, and get right into this great discussion with Andrew. Okay, I'll leave it. Oh, it looks like we have the majority of our audience have cast their vote. So thank you very much for that. But I see there's a couple more people out there that are just now trying to place their votes. So go right ahead. I'll leave it open for a few more seconds just to get you involved too. Perfect. Thank you very much, everyone. Okay, so it looks like we got the majority of our audience here, and those few other people have now made their selection. So I'm now going to close the polls, and let's take a look at the results. Okay, so here are the results, Andrew. We have in our audience 28% said quality and compliance issue, while 12% said efficiency issues. Then 12% again said variability between devices and or operators. And then surprising, nobody said cost issues. But then we have 48% of our audience that chose all of the above. So thank you very much, everyone, for participating in our first poll question. And now with the poll is out of the way, Andrew, before you get into your discussion, What do you think about the results of that poll question? Anything surprising there?
Yeah, I think cost is always an issue for folks, but I'm
Yeah.
sure a lot of that got rolled into that 48%. I think a big component of, liquid handling in the lab is, this involves a lot of expensive reagents, expensive labor, expensive instrumentation. So getting the bang for your buck out of it is, always a ever-present concern.
Okay. Thank you very much for sharing that. And with that, I'm going to pass you the mic and the controls. And when you're ready, you may begin.
Thanks. So yeah, the title of my talk is Accurate Liquid Handling in the Lab Using the Multi-Channel Verification System, or as we call it, the MVS. So a little background for me. I am the field application scientist for the West Coast for advanced instruments, focused primarily on the Artel portfolio, which is our liquid handling validation. So I've been in the industry for about 20 years, started in academia doing small molecule drug discovery, then switched over to doing a lot of automated analytics, cell and gene therapy manufacturing, small and large molecule discovery across quite a few different companies in small and large biopharma. But the one consistent thread throughout my career is that foundationally accurate and traceable liquid handling is paramount. It's all that we do. So the Artel technology works in that space. So we employ a dual dye, dual wavelength, ratiometric, absorbance-based measurement. So you can get into it with the general chemistry theory behind it, which is relies pretty heavily on the Beer-Lambert equation. So we just take two absorbance peaks and then measure the ratio between the two, and then the whole process takes about 10 minutes, but you get very, very high quality measurements of accuracy and precision. So here's the MVS itself. It's got a plate reader connected to a laptop, and then it comes with all the accessories, the dyes, our characterized verification plates, which are a very important part. All of our reagents are barcoded for traceability purposes. And then we have a shaker to ensure that the sample is adequately mixed to get you your best possible results. So the basic process, we dispense the red dye into our verification plate. We add the blue diluent. This step can actually be reversed depending on what's closer to your actual real world process. Shake the plate on our plate shaker with verified settings and speeds, reads the plate, and you get your report. The reports are really nice because they have kind of a wealth of data for statistics for the full plate, accuracy by channel, and dispensing order for replicates. So this is kind of what it looks like. You get a heat map, some statistics up on top for relative inaccuracy, coefficient of variation, and then a pass-fail criteria set by the user. So you can set your own tolerances for your reads. So the MVS has quite a few, use cases and methodologies. An obvious one is troubleshooting your assays or equipment, checking reproducibility for samples, mother-daughter plates. It's also really good for spot checks if you want to make sure the instrument's performing correctly after a crash, after service and IQ/OQ, returning it to certified usable states. You can use it to monitor critical target volumes for assays. I know a lot of multi-omic assays can have some really expensive reagents involved. So being able to use those appropriately is really important. You can also use the MVS for validating after site acceptance testing. A lot of our customers are actually the manufacturers of a lot of laboratory instrumentation, so a lot of them use the MVS to as part of their QC departments to make sure their equipment's working properly after manufacturing. We can also do some proficiency testing for multi-channel pipettes. You would be amazed with the wide variety of pipetting skills you find in the lab. There's no real, a lot of places don't really, put it in their job interviews or anything, and a lot of people learned a lot of bad habits over the years. So being able to get everybody on the same page is really important. And the last one is it's great to validate results for scaling up methods or doing tech transfers to other organizations or groups in your lab. The other nice thing about the Artel portfolio is we conform to all sorts of regulatory standards for many different fields. We're traceable to NIST, USP for pharmaceutical, a lot of manufacturing specs, and then ISO and CAP-CLIA for the clinical markets. The other, critically important thing the MVS does is it can compare liquid handlers and people. So device to device in a lab, when you bring in the MVS, you immediately start to see, you've got a pretty good variation between instruments. No two instruments are exactly the same. So being able to have that, direct, reliable comparison is really important. And the other great thing about our process is it's standardized. So if you're a global organization, you can, be confident in your results that, 10 microliters in France is the same 10 microliters in Boston. Also, yeah, like I said earlier, you can have two of the same instruments programmed exactly the same, but due to manufacturing issues or relative health of the instrument itself, there can be a lot of variance in your results that you might not even know of. And in really important use cases like, say, forensics, this kind of data is really important because if you get it wrong, a whole lot of legal work and things can change someone's life, and probably not for the better. You can also, like I said earlier, we can directly compare operators. So everybody's different, and being able to see the inaccuracies and get everybody on the same page is pretty important. And then, so this is actually a great set of slides. The PCS is our handheld pipetting system, and we took data from 475 trainees doing 5 replicates of a 20 microliter transfer. And this is pre-training. You can see there's quite a bit of clustering, but there's also a ton of outliers. And after everybody's trained and on the same page and using the appropriate techniques, that all goes away. And so the optimization isn't just limited to manual pipetting. You can also use the system to lock in both accuracy and precision for the instrumentation. So just a quick reminder, I love this little dartboard diagram. I think it's a really elegant way to show accuracy and precision. But so precise means all of our data points are close together and repeatable. But accuracy is how close they are to the target. So you can be accurate and precise. You can also be not accurate, not precise, or any combination in the middle. So the first use case that I used to use routinely with the MVS was optimizing liquid classes. So when we say liquid class, we mean the settings on an automated liquid handler to pipette, taking advantage of speeds, blowing out the pipettes, pre-wetting tips, pausing to allow the material to transfer through the pipette tip, and then aspirate and dispense heights. So different liquids are going to act differently. Pipetting ethanol is going to be different than pipetting, serums, NGS reagents, that kind of thing. So being able to tune the liquid handler to your environment, your material, your tips, and everything really affects your results downstream. And so we have a process we use called DMAA, or Dispense, Measure, Assess, Adjust. And it's basically a way to systematically go through the settings and tweak them based on the results you're getting for relative inaccuracy and coefficient of variability. And then by iterating through all these different steps, you can really dial in the results. As you can see on the bottom there, almost 8.5% inaccuracy and 5% CV can turn into 0.08 and 0.03. And getting accuracy with the MVS under 1.1% is actually pretty easy. There's even software out there from companies like Synthase that do design of experiment-based optimization. So based on the data you get, you can feed it back into their software and get the next group of testing parameters, which is really nice for optimizing parameters. I wanted to just call out this part. I did a lot of IC50s and dose response plates where you use a lot of serial dilutions, and amounts of these compounds are put into activity assays, DMPK, SAR, that kind of thing. And knowing how much you're actually getting in your compound in a 40 microliter assay volume, even if it's, you think you're getting 10, but you're getting almost 7, it can be a huge difference in your compound concentration. So being off by, 3 microliters ends up being nearly a 40% difference in concentration. And that's something you really want to avoid, because that's a variance you don't really know about until you can accurately measure it. And even on the other end, in the diluent, just plus or minus 4 microliters adds up to a 57% difference in compound concentration. So that really skews, IC50 curves and downstream data, which can cause a lot of problems. We wrote a whole paper about this. It's pretty interesting. I believe, it should be on our webpage, and it's been published in Gen. The second one I wanted to do was a really, I want to say kind of out of the box way to use really high level pipetting data, and then use that to drive decisions in the lab. One of the things about laboratory automation is it's really expensive capital equipment. And then, once you buy that, there's also, some additional expenses and, labor and consumables, but there's also, service agreements for keeping them in good shape. And some of those can get really expensive. And as the instrument ages, there's ways to, save money on service by going for, parts only or preventative maintenance only on kind of less critical instruments. You can go with third parties. They can be expensive, but they don't have they don't always have the right certifications for ISO and NIST traceability. Well, the MVS does both. And so where I was in a large biopharma lab where we had 7 big integrated systems and a total of 40 liquid handlers, a wide variety of dispensers, automated liquid handling robots, plate washers, that sort of thing. It was a very mature lab that's been around for about 20 years, so there were quite a few different instruments used in a lot of different modalities. It was a core facility, so people could walk up and use instrumentation. So seeing how getting a wide picture to see kind of the bird's eye view of how the whole lab was performing was really important. So going back, we did our change management for the service agreements to follow our assess, design, execute, and sustain strategy. So we took the data from the MVS validations for all the instruments and then combined them with the usage log of the instrument and our records of who was running what methods, running what experiments on which system, what their failure rate was, how many service calls we had to provide. And then we started triaging the instrumentation by essentially criticality. If it was something that, we could rerun if there was an issue, or something that absolutely had to succeed, and or was walk away, lights out automation. That's something you want to have a really high level of uptime with. And then from there, we were able to kind of figure out which instruments we could get away with lowering the service contract costs on. So some of them, they were running some walk-up stuff that, if there was an issue, we could, use redundant instrumentation, have the, it wouldn't be a huge problem if an instrument was down for a couple of days, since we had plenty of redundancy. And so we were able to get some pretty significant cost savings from that. So our critical path stuff, like the integrated systems, all remained on full contracts. And then walk-up systems, we just decided to deploy walk-up systems based on how well they were used. And then we were able to find a couple problem instruments that were the lemons, some older instruments that weren't really getting a lot of use or weren't really performing up to our expectations. So we could retire them or trade them in. And all in all, that saved about $300,000. So it was kind of a testament to the core technology to see, like, okay, when you get this amount of confidence in your results, you can make a lot of decisions to use your lab equipment more efficiently. And yeah, so that's my conclusion for that use case. The dual die ratiometric verification is pretty much the most certainty you're ever going to have in the lab. I know we're always working with theories, hypotheses, and a lot of known unknowns and unknown unknowns. And the MVS really just solves the whole problem in one package, which when you really start to understand that, you can do a lot of performance management, asset management, and make a lot of good decisions in your lab. And with that, I'm going to hand things back. Thank you so much for having me on.
All right, Andrew, thank you so much for that insightful information with regards to the automated side of what happens in laboratories. I'm now going to just kind of swap over a little bit and talk a little bit about what about those manual handheld pipettes. There's a lot of myths that happen in the laboratory when it comes to pipettes. So the first thing that I want everyone to consider, right, and just kind of think about in your brains, if you will, just quietly, is, can anyone pipette? Can anyone pick up a pipette, and can anyone use a pipette? And then more importantly, that same question with a few words that are added is, can anyone pipette accurately and precisely? So same question, but a much more deeper dive into, like, what does it really mean? And here's what we know. We've trained hundreds of scientists and analysts through the years with our pipetting technique certification courses. And this is an example of the data that we see for the pre-training data. We come into a laboratory and we assess people with, how are your pipetting technique skills today, right now? And as you can see from this data, while there's a large group of people that can pipette accurately and precisely, there's also a pretty significant amount of people who don't pipette as accurately and precisely as they could. So I want you to just think about, if you were to assess all of the pipette operators in your laboratory, or in all of your laboratories, where would they fall in this data chart? We know quite a bit about the things that impact how a pipette performs. Operator technique is definitely the largest source of variability when it comes to pipette performance. If you take a look at this pie chart, accidental failures is the next to the largest source of variability when it comes to pipette performance. And guess who causes all of that? We do. Did you look in the mirror today? Your beautiful face causes the significant, percent of what happens when these pipettes aren't working well in the laboratory. It's a pretty well documented topic, this topic of what happens when you've got people using pipettes, and there's a lot of variability with how they're using them. So much so that there's a set of standards, ISO 8655, that all has to do with the pipette world and the volumetric apparatus world, if you will, and pipettes certainly fall in that realm. And if you take a look at this, in Annex B of ISO 8655 Part 2, they specifically call out how much impact pipetting technique variability can cause. And so it can cause up to an 8% impact on your volumes. So imagine if you're looking for something as small as 2 microliters, an 8% impact on 2 microliters can be pretty significant. And to Andrew's point, it can really take an assay and really change the outcome. So what does that really look like at the end of the day? We're going to explore 2 of the techniques that are on this list specifically. So the first technique that we're going to talk about is tip pre-wetting. So hopefully pre-wetting is a technique that a lot of you are familiar with. And what I'd like to do is I'd like to play this next slide. And I want you to take a look at this video for me. And as you watch this video, I want you to think about, is this how you pre-wet your pipette tips? Is this how you see operators in the laboratory pre-wet their pipette tips? I'm going to take a moment and think, is that what you see? Is that what you observe? Is that what you do? And in fact, this technique that was just played in this video is what we call the imposter. That is not how you properly pre-wet a pipette tip. Now, some of you might be thinking, oh my gosh, really? This is how I've always been doing it. I've been doing it this way for years. And in fact, right, when this particular technique is called rinsing or priming, and in some worlds it's called mixing, right? And so what ends up happening when you place your pipette tip into the sample and you simply bounce that plunger up and down, it puts you at risk for a few different things. It puts you at risk for bubble development. It puts you at risk for the sample to shoot up inside of your pipette tip to the point of no return, right, that you can't get it out. And in addition, now you're operating the pipette plunger in a way that it's not meant to be intended to be operated. The pipette plunger, if you're using it in forward mode with aqueous solutions, is meant to start from its home position down to first stop, to pull its aspirate, and then go ahead and make your dispense sequence. So let's take a look at what that truly looks like in just a moment. Now on this slide, first we need to kind of dial it back a little bit and think about, when should we be pre-wetting pipette tips? And the answer to that question is every time you put a new tip on your pipette. Here's what happens, especially those of you that are in colder climates in the wintertime, the heat is on, your laboratories are probably getting really dry, and you can feel that because your lips are getting chapped. The skin on your hands are getting chapped. You can see how dry everything is, right? When you put a new tip onto a pipette and you go and you pull your initial aspirate of that sample, evaporation starts happening. And evaporation starts happening because that airspace is not humidified. And the whole purpose of pre-wetting is to humidify the airspace above your sample so that you don't see sample evaporation occurring. And how you do that is to aspirate and fully expel a minimum of 3 times. And we're going to talk about that minimum of 3 times recommendation in just a moment. But first, let's take a look at how you properly pre-wet a pipette tip. So in this video, you can see that we've got the plunger pressed down to first stop. We place the tip into the sample. We release the plunger. We remove the tip from the sample. And then we place that tip against the sidewall, and we fully press that plunger all the way down to its second stop. So that's how you properly pre-wet a pipette tip when you're using forward mode. And you usually are using forward mode when you're working with aqueous solutions, right? So again, a full aspirate and a full dispense is how you properly pre-wet pipette tips. So you might be wondering, where does that minimum of 3 times come from, right? Because sometimes you have to do a little bit more than pre-wetting 3 times. If you take a look at this particular data set, right, I've got 3 different 20 microliter pipettes. The very first 20 microliter pipette uses a 200 microliter pipette tip. And if you take a look at the data, what I did is I put a new pipette tip onto the pipette, and I immediately started taking data. And you can see that that rule of 3 really stuck, right? You can see that the first data point, 19.75, was a little bit low. Second one got a little bit better. And then the third and the fourth data point, that's really when that airspace started to humidify and the volumes started stabilizing, right? So for that pipette and tip, right, a minimum of 3 times was pretty sufficient. If you look at the pipette that's in the middle of this graphic or of this slide, this is also a 20 microliter pipette that is also using a 200 microliter tip. But you can see there's a little bit different data that's occurring here. In fact, 5 pre-wets was needed for this particular pipette and tip system. 5 pre-wets is when we started to see that sample volume stabilize, that airspace started to humidify, and we started to see volumes closer to our target of 20 microliters. Now this very last pipette, in some of my training, some people refer to this pipette as a cheater pipette. In fact, it's just designed a little bit differently, right? You can see that the airspace is really minimal in this pipette tip when it's placed onto this pipette. And in fact, if you look at the data, one could argue that 19.98 is the same as 19.99, not much different than 20.01. And in this particular set of pipette and tip arrangement, right, a single pre-wet is probably all that you needed in that scenario. So what's the purpose of this slide, right? From a really high summary, the larger the airspace, the more pre-wetting that has to occur. So if you're using an adjustable volume pipette and you've dialed it down away from its highest volume and that airspace is increasing, more pre-wetting is going to be necessary. Otherwise, you're going to see a lot of sample evaporation happening. The second technique that we're going to talk about is pipette tip immersion depth. So we've got a poll question. So the poll question is, if you are aspirating 20 microliters, what is the proper tip immersion depth into the sample vessel prior to you aspirating that sample? So Sonia, I'm going to ask for your help with managing these poll results, please.
Okay, and I just launched the polls. If you want to give them their options they can select from, go right ahead, Candy.
Excellent, thank you. All right, so your options are 10 millimeters, 6 millimeters, 4 millimeters, 2 millimeters, or between 2 and 10 millimeters. So we'll give you about 45 seconds to go ahead and make your selections, and Sonia's going to see how everyone's doing with their responses.
Oh, well, right now we've got less than half of our audience members, so I think they're all trying to make their selection now. So just encouraging everyone that remember this is done in real time. So if you can just go ahead and place your vote now, then I'll close the polls in a few seconds, and then we will get right back to Candy and her thoughts on the results.
Thank you,
Okay,
Sonia.
Candy. Oh, look, it looks like we got the majority. More than half of our audience has now jumped in, leaving it open for a few more seconds just to get a couple more people involved here. And okay, that should do it. Thank you so much, everyone, for participating in poll question number 2. I'm going to close the polls now, and let's take a look at the results. Okay, here we go, Candy. We have in our audience 24% said 10 millimeter, while 3% said 6 millimeters. Another 10% said 4 millimeters. We got a whopping 45% of our audience that said 2 millimeters, and then 17% said between 2 to 10 millimeters. So here we go. What are your thoughts on the results of that poll question?
Excellent. Thank you so much, Sonia. Bear with me. I'm going to resume the slideshow. All right. So here's the good news. Those of you that had guessed 2 millimeters, you are absolutely correct. You deserve a golden star today, right? 2 millimeters is the correct answer when you're aspirating 20 microliters. And 2 millimeters is really not very deep at all, right? Certainly the tip immersion depth does vary based on the volume that you're working with. And believe it or not, 6 millimeters should be the deepest that you should ever go when it comes to aspirating and aspirating your sample volume. So let's go ahead and take a look at that, right? So we have some recommendations from both the ISO world and the ASTM standards, right? Again, as I mentioned, 2 to 6 millimeters is the kind of the range, if you will. Depends on the volume that's being aspirated. And you might be wondering, well, why is that? What happens if I place my tip too deep into the sample? Well, 2 things happen if you place your tip too deep into the sample. The first thing that happens is now you have more surface area for drops to cling to. So if you're working with your sample volumes and you've aspirated your sample and you see droplets on the outside wall of your pipette tip, some of that might be attributed to the fact that your tip immersion depth is just simply too deep, right? And then the other thing that happens is not only does a deep immersion depth put you at risk for droplets on the outside wall, but it also, the forces of the pressures on the outside of that pipette tip forces too much sample into the tip. So it's almost a double whammy, right? You definitely over aspirate your volume, and maybe that's not too much of a concern. But of course, then the opposite effect happens is if your tip immersion depth is too shallow, then it puts you at risk for aspirating air. And oftentimes, under aspirating the volume is definitely not something that most folks want to have happen. So to put it into a little bit of relation to what does that really look like, this particular graphic is a pipette tip that has been marked at 2 millimeters and 6 millimeters that's set beside a quarter, right? So 2 millimeters is not very deep. It's breaking the surface of the meniscus and maybe going slightly a little bit deeper. And 6 millimeters is not very deep either, and that's really set aside for 10,000 microliters. That's a really big volume. So what you may find yourself doing, and likely doing, if you are working with those larger volumes to maintain that 6 millimeter immersion depth, you may find that, that you have to follow the sample down while maintaining the tip immersion depth, and sometimes that takes a special degree of attention for sure, maybe even a little bit of wizardry that happens. So again, that really deep immersion depth puts you at risk for drops on the outside wall of the pipette tip. The shallow immersion depth puts you at risk for bubbles at the bottom, right? And that bubble represents where sample should be, and that means that you've underestimated your volume. So let's take a look at the data. What does the data look like? So in this particular data set, our target was 20 microliters. We're placing our tip 8 millimeters into the sample. You can see that we've got some spikes in data. So those are droplets, or what I like to call Klingons, on the outside wall of the pipette tip. So for those of you that are Star Trek fans, you can identify with that, right? And when we take a look at this next data set, it's a 1 millimeter tip immersion. It's a little bit too shallow. You can see that we're aspirating some air. Instead of aspirating 20 microliters, we're aspirating about 19.75. We're definitely not getting that full sample volume amount. And then last but not least, our last data set that we have is kind of like Goldilocks and the Three Bears, right? The 2 millimeter tip immersion depth is the sweet spot. That's exactly where we want. All right, so we have another question, and this time it's not going to be in a poll format. It's just open for you to enter your answers into the chat box. And I want you to take a look at this graphic and just enter into the chat box, what do you see that's missing? So again, as you take a look at the graphic that's on this slide, what's missing? So go ahead and take a moment or two, enter your answer into that chat box, and then we'll talk about what's missing on this slide.
And just as a reminder to our audience members, that chat box is located on the right-hand side of your screen.
So yeah. Okay, so hopefully those of you that wanted to answer the question on this slide about what's missing, you've entered your answers into the chat for us. All right, so let's talk about what's missing. So first and foremost, those of you that answered gloves are missing. Yes, that's often a critical PPE component of anyone that's working in the laboratory. But those of you that answered the pipette tip is missing, that is the one thing that is like, whew, that's definitely probably the more correct answer. Really, both gloves and pipette tip is correct. It's interesting, right? We have seen in some of our pipette technique classes, our certification classes, where when people pick up the pipette and they go to use it and they forget to put a tip on, but that's okay. I chalk it up to just some nervousness, right? Because now you've got someone watching your pipetting technique. So what's the purpose of this slide? A lot of you, if not all of you, have heard the saying, don't believe everything you see on the internet. That's what the purpose of this slide is. So this is a graphic that we've taken from the internet, right? And just because you see recommendations about pipetting or how to handle pipettes or manage pipettes on the internet doesn't mean that it's correct. And so definitely be aware, take caution with the information that you're seeing. And more importantly, please remember to put tips onto your pipettes when you're using them. But truly, I hope that you are as content as she is when you're doing your work in the laboratory, because she seems pretty content with the work that she's doing for sure. All right, so now let's talk a little bit about, okay, pipette operators and the requirements for assessment of pipette operators. So I have two different standards that I've mentioned on this particular slide, ISO 8655 Part 10, as well as ASTM E1154. They both mention training and testing your operators, so that's just a given. When you're onboarding someone and you're training them to perform certain assays, training's going to happen, right? But you need to have demonstrated competence of those users. But then there's another part that both of these standards mention, and that's retraining and retesting. So that's consistently assessing pipette operators a minimum of every 12 months. You need to have those records of competency on hand when the auditors come, when the inspectors come, and they ask you, how do we know that all 50 analysts on the bench are pipetting and using pipettes correctly? It's your responsibility as the lab manager or the person that oversees all of them to be able to provide those records of competency at least every 12 months. And I encourage you to have fun with it. You can see in the upper left-hand corner, we've got like a little image of medals. Part of our certification courses is to award medals to people based on their performance in the practical portion of our trainings. They have to be able to pipette well. And a lot of them earn gold medals because they're definitely paying attention. They've refined some techniques, and now they're successfully pipetting with precisely and accurately to the best of their ability. There's nothing better from a trainer's standpoint to see that happen. All right, so let's talk a little bit about some of the ways that we can help. So we have a PCS instrument that's pictured here in the lower right-hand corner. PCS stands for Pipette Calibration System, but in fact, this system is that and a lot more. Certainly, if you wanted to calibrate pipettes in-house, you could certainly do that with the system. But more importantly, and related to everything that I've spoken about, there's also a built-in operator assessment module built within the system. And so I have a sample operator training report that you see here. This could be part of the training reports that you provide to auditors and inspectors when they ask you for those records of competency. The great thing about the PCS is it allows you to assess your operators in less than 4 minutes. So it's really fast, and you can assess operators from 0.1 microliters up to 5,000 microliters. You can perform trainer-led assessments, or operators can perform self-assessments. You can also enable automated email notifications when those operator assessments are due. And last but not least, if you wanted to manage all of that, pipette calibration, operator assessments, if you wanted to manage everything digitally and not have any paper in your laboratory, the application, the Artelware application that works with the PCS, enables you to be completely digital for review and approval workflow, as well as for results. The fact is paying attention to the details pays off. So you might recall earlier on, we had some pre-training data that I showed from years and years of our certification courses, and this is that post-training data. No matter how long you've been on the bench pipetting, I've met people who've been pipetting for decades, and they've always said, oh my gosh, I wish I had taken this training back at the beginning of my career. The fact is a lot of people just don't know what they don't know. And then when they take part in our courses, they understand the how, they understand the why, they understand what happens to the data if they don't. And their eyes, they have like an aha moment. And again, that's really some of my favorite parts of our training sessions for sure. But then there are some things you can't control. So we're going to spend a little bit of time talking about the things you can't control. So first and foremost, the environmental impact. So ISO 8655, they also spent some time talking about what causes variability with air displacement pipette performance. Atmospheric pressure certainly has an impact, right? If you're in California, you probably are not going to send your pipettes to Colorado to be calibrated for some obvious reasons. Temperature has an impact. If you think about what temperature does when it's warm, when it's cold, it contracts, it expands, right? That certainly has an impact on these air displacement volumes. Relative humidity has the largest impact. We talked about tip pre-wetting. If your laboratory is really dry in the wintertime, you're going to have sample evaporation occurring. So just from the environment itself, you could have a 3.5% influence on the volumes. And if you add that to the 8% influence from your operators, you can see that that really kind of compounds quickly, right? Now you're up to an 11.5% potential for error on your volume. So that's very eye-opening, okay? So how do we help? We've got so many resources. We've got a number of posters. We've got posters to help aid with pipetting aqueous solutions. We have posters that help aid with pipetting viscous solutions because, of course, they move slowly. They handle differently, right? We've got lab reports. We've got videos. We've got a number of different resources that can help with regards to making sure that your pipette operators are educated. And more importantly, I've got an upcoming class that's coming in October that's occurring here in Maine on October 16th through the 17th. It's a 2-day training program dedicated for those of you that might be overseeing the whole pipette management program at your facility. So pipette management program includes pipette calibration, pipette verification, pipette repair and maintenance, your compliance to various standards and regulations, and then also like training your operators, making sure that you're assessing them. And so if you're interested in coming to Maine and you're interested in learning more about how to manage that pipette program in-house, I would love to host you in October. It's a beautiful time in the state of Maine. All right, last but not least, it's time for our final poll question. And Sonia, I'm going to ask for your assistance with this, please.
Certainly, and I will launch the poll question now, and audience members, you should see it appear. This is our final poll question, and it's all about you. The results won't be shared because we'll get right into the Q&A portion of the webinar. And as a reminder to our audience, you still have the opportunity right now to send in your questions using that questions window. And for those of you watching on LinkedIn, please send in your questions using the comments tab. Okay, so you have here a couple of options below. I am most interested in, is it a PCS, Pipette Calibration System, MVS, Multi-Channel Verification System, or Pipette Certification Training? So I'll leave this open for a few more seconds just to get everyone involved. So go right ahead and make your selection, and we'll wait here patiently for everyone to cast their vote, and then we'll get into the Q&A. Okay, that looks like it's half of our audience. Now, if I can encourage the remainder to go right ahead and make your selection, that'd be greatly appreciated, everyone. Okay, perfect. Thank you so much, everyone, for participating in our final poll. I'm now going to close the polls, and then we'll get right into the Q&A portion of the webinar. So let me just take the controls from you. Okay, and then we'll join on camera with Candy. So I'll have Andrew join with me. Hello, everyone, and Andrew will join us. And let's look at the questions that have come in. So first of all, I'd like to say thank you very much for that very engaging presentation. It was fantastic, and I'm sure everyone appreciated that. So let's get into our first question here. And let me just see where that is. Here we go. So this audience member is asking, what types of pipette-related certification programs do you offer? So they want a little more clarification. So if you can list them, they'd be greatly appreciated.
Excellent. Thank you so much, Sonia, for that question, and thank you for the audience member that posed that question. As I mentioned on one of my previous slides, we have a couple of different flavors, if you will, of our pipette certification classes. The first one is a pipette technique certification class, and that's where we come on site to your facility and we certify your analyst on pipetting technique. And you're then provided with a data summary of as found data versus as left data so that you can see, exactly kind of your return on investment, if you will. And we also provide a lot of additional information in that data packet as well. So that's the pipetting technique certification that happens on site at your facility. And it's really geared more towards certifying the folks that are on the bench. And then the other pipette related certification class that I have, the one that's coming up here in October, that pipette quality management certification, that's really geared toward lab managers or lab supervisors or metrology departments that are responsible for overseeing the pipette program that's occurring in-house as a whole. So really two different types of programs, and certainly if you're interested, reach out to us. We're more than happy to talk about the different types of modules. We have a single channel module, a multi-channel module, a viscous solution module, pipette repair and maintenance module, lots of different options depending on what it is you're looking for.
Oh, thank you so much for going into so much detail with that. Thank you. All right, we'll go to the next question here. This audience member is asking, what type of sample solutions are available with the MVS system? Who would like to take that question?
I can take that one.
Thank you, Andrew.
So we have our traditional aqueous qualiture solutions in a bunch of different ranges, and the ranges are concentrations to fit the range of the reader for certain volumes. So if you're testing a kind of nominal volume in the lab, anywhere from 50 to 200 microliters, we have our range A. Below that, we use the range B, and it goes all the way down to range F. So that's for single-digit nanoliter dispenses from like an acoustic liquid handler, like an Echo. We also have additional formulations of the dye for NGS-based approaches. We have a PCR mix solution that behaves a little bit more like PCR reagents. We have one for plasma and serum, which are generally used in clinical applications or biomarker assays, patient samples, that kind of thing. And we also have a DMSO-based solution for a lot of compound management groups for small and large molecule drug discovery, and for dedicated users, we actually have a solution where we can use our dye stock to mix with the user's own material. So if you really want to put the extra work in, you can get a dye solution that matches the real world contents of what you're trying to pipette accurately. And that's I've been able to deploy that with some pretty good results.
Oh, fantastic. Thank you so much for that, Andrew. Let me jump to the next question here. This audience member is asking, can your systems be used to perform both calibrations and verifications? Another great question. Who's jumping in on that one?
I'll jump in on that one. The answer is they can be used to perform both. And so we have some customers that will use our systems to perform their semi-annual or annual pipette calibrations or automated liquid handler verifications, right? Depending on the world that you're in, calibration and verification might be synonymous in term, or they might mean different things, right? Sometimes people think calibration means you're doing something physically to the pipette, but that's not always the case in a lot of other worlds. And then we also have some customers who purchase our systems to perform interim verifications. So if you are getting your devices calibrated or verified annually, but you still want to know throughout the year, is it still staying in calibration? Is it still in specification? Then we also have some customers who will purchase the systems to perform those interim verification checks. We have some customers that perform them daily, like our forensic labs, which make us very excited that happens, right, for DNA analysis. And then we have some customers who maybe perform monthly or quarterly verification checks, but definitely some additional checks, again, just to make sure that their devices are staying in specification, staying in calibration, if you will.
Mm-hmm. Thank you, Candy. All right, let's looks like it might be our final question here as I'm looking at the time. So this audience member is asking, if my laboratory is ISO 17025 accredited, can we use your systems to verify performance of our pipettes and liquid handlers? Who is going to take that question?
I can take that one.
Thank you,
the answer is yes.
Yes.
Our process conforms to that specific ISO standard 17025, which I believe is for handheld pipettes. Candy, is that correct?
Oh, sure. So 17025 is really more about it could be anything that needs calibration. And more often than not, our 17025 accredited laboratories will follow the ISO 8655 standards. Really, they say what or the how. 17025 says what you should do. 8655 says here's how you should do it, which is always nice. Sometimes people just want a roadmap. How should I do it? So you're absolutely right, Andrew. If you are a 17025 accredited laboratory, our standards, or rather our technology, is mentioned in the ISO standards that are typically used for pipette calibration and automated liquid handler verification in laboratories. And 23783 is the other ISO standard,
Yes.
but more so for automated liquid handlers. Lots of numbers. I know I'm a geek because I can rattle those off. I'm so sorry.
I'm just amazed.
Long story short, the Artel process is kind of like the Staples easy button for regulatory compliance. If you just use it, it's a solved problem.
I like that answer. Thank you very much, Candy and Andrew, for answering all those questions. I know we have a lot more questions that are lined up, but unfortunately, we've come to the end of the Q&A portion of the webinar. So rest assured that the team at Advanced Instruments will follow up with you after this presentation. And if you have any further questions, please direct them to the email addresses you see there on your screen. Okay, thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our webinars. Additionally, give me a few seconds. I'm going to share a link to view the recording of today's event in the chat box, which you can also share with your colleagues once they register for the recording as well. And for those of you watching on LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. And there is more. Get more out of today's presentation by downloading supporting materials that we have there available at the Handouts tab. And we have two there for you, Pipette Calibration System and Multi-Channel Verification System. So please go ahead and check that out. Now, please join us in thanking our speakers for their time here today. So thank you very much, Andrew and Candy. It was fantastic.
Yeah.
We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. Until we meet again, I'm Sonia Hunt. Bye for now, everyone. Take care. Bye.